Bismillahirrahmanirrahim. Assalamu alaikum everyone. My name is Ayesha Gull and I am the student of M.phil Microbiology second semester from Cohort University of Science and Technology. The topic of my presentation is isolation, identification and characterization of potential oil degrading bacteria from oil contaminated sites. This is the research paper which gave the concept of such topic. This research paper was published in Journal of International Research Journal of Engineering and Technology. These are the outlines of my presentation, which are introduction, objectives, methodology, result and discussion, conclusions, further recommendations, and references. Let's start with introduction. In this modern world, the need for petroleum products had highly increased for the contribution of modern lifestyle. Petroleum products are complex mixture which are mainly derived from crude oil and they are processed in oil refineries. Gasoline and diesel are some of the petroleum products refined with the high allocation of energy from the oil refineries. The hydrocarbons containing oil like crude oil, petrol, diesel, etc. cause environmental pollution. pH media was used as enrichment media for the isolation of diesel oil degrading bacteria. Hydrocarbons found in crude oil are metabolized by a large number of bacterial communities into non-toxic biodegradable product. The release of such petroleum product into the environment shows a direct impact on biotic and abiotic communities of that environment if not treated at a time. Let's talk about materials and method. In methods, we will follow some steps. Step 1, sterilization. Step 2, sample collection. Step 3, enrichment of microorganism. Step 4, screening and isolation of oil degrading bacteria. Step 5, measurement of bacterial growth. Step 6 is identification of bacterial isolates. In this we will do staining, biochemical test, and step 7 is oil degrading studies. Now let's start with materials. In materials we will use media containing Bushnell has broth, Bushnell has agar, nutrient broth, starch hydrolysis media, tryptone growth, carbohydrate utilization media, and methyl red broth. We will also use autoclave, oil contaminated soil samples, and incubator. Now let's come on result and discussion. It shows some results like enrichment of microbes. The microbial culture were enriched for oil degradation using pH media with engine oil as sole carbon source, and it was confirmed by comparing the flask before and after enrichment of microbes. Flask A shows before the enrichment of microbes and flask B shows after enrichment of microbes. Screening of oil degrading bacteria. Enriched culture of 2 soil samples were sprayed on the beach agar plate, which is the selective media for the oil degrading microbes. The oil degrading bacteria were grown on the media, which is compared to the control plate. Isolation of oil degrading bacteria. Here we isolate oil degrading bacteria. Biochemical test. In this we do catalase test. Isolate 1 and 2 are negative for the catalase test. Isolate 3 and 4 are positive for catalase test. Indole test. Isolate 1 and 2 are indole negative. Isolate 3 and 4 are indole positive. Methyl red test. Isolate 1 and 2 are MR positive. Isolate 3 and 4 are MR negative. Starch hydrolysis test. All 4 isolates are positive for the starch hydrolysis and it was confirmed by clear zone formation. Oil degradation. 4 isolates bacteria were allowed to undergo oil degradation of petrol, diesel, and using engine oil for 20 days. Here are some results. Degradation of oil. A show before and B show before degradation of oil. Gravimetric analysis. Gravimetric analysis was done to extract the oil degraded by bacterial culture using the petroleum, ether, and acetone in ratio 1:1 of 20 ml. Now let's come on conclusion. From this study, it was concluded that the oil contaminated soil is the best source for the isolating of oil degrading bacteria and may be used for the isolation of other microorganisms. The isolated bacteria can be used for the degradation of different hydrocarbons containing oils, and it can also be used for the remediation of contaminated sites. These are some future prospects. Checking for the degradation of other organic compounds like chemical fertilizers with the isolated bacteria. Also for the production of biosurfactant using the isolated bacterial strands. These are some references. Thank you so much for your kind attention. If you have any question, then you can ask me in the comment box. Thank you so much. Allah Hafiz.